I'll start with just a really basic review. It's a really complex topic, but essentially, if you look at like the retina on the back of your eye, there's a million fibers coming off the retina, and they project to the thalamus, and then the thalamus projects to the cortex. See the area in the cortex is V1, and then this basic arrangement holds true also for auditory and somatosensory input. So everything from your senses goes to the thalamus before it gets to the cortex. And it also holds true from regions of the cortex projecting the region of the cortex. They also go through different regions of the thalamus. So the thalamus has a bunch of little regions, each one dedicated to certain things, but it has this very common and simple structure, which no one understands what it does at all. But we have a theory about it. Here's a green orange thing here. Relevant to today's discussion, there's two, if you look in the cortex, you look at a column in the cortex. This is now layers 1 through 6 in a sheet of cells. And if this is in B1, there's two projections back. There's 6B and 6A, and they both project back to the thalamus. And so in theory, these are large projections. In theory, the cortex is telling the thalamus to do something about the information that's being sent to the cortex. And for almost all the literature on the thalamus, that is just, it's like they have no idea what it's doing. They'll say, oh, it's changing the receptor field a little bit, timing a little bit, size of, it doesn't look like it's doing anything. And they refer to the cells in here as relay cells because for a long time when they observed them, once it would be like a 1-to-1 ratio coming in. You have like about 1 million fibers coming in, you have about 1 million fibers coming out. You get a spike on one of these cells and you get one single spike out here. Now that's not true, but that's true in some situations, and they observed that. And so they call them relay cells because they didn't seem to be doing anything. A spike comes in, a spike comes out. But this thalamus is critical to everything in the brain. And if you don't have it, you're dead, and or you're a vegetable. And so, clearly that didn't look right. But that's how people talk about it, and that's why I talk about relations. Okay, so, well, leaving out a lot of details, we have hypothesized that one of the major functions of the thalamus is it does orientation transforms. So you have an input coming from the retina, for example, and then you rotate the eye relative to the world. That whole, all the inputs are rotated, and you have to sort of reorient it back to the canonical form. And the same thing is true with motion. That is, if your eyes are moving, if you're reading text and you're reading from line to line, my eyes horizontally to read the text this way, but if the page is slightly tilted this way, I can still read, but my eyes are no longer moving horizontally, they're moving like this. And so you have to compensate for that because the models in the cortex are not, they're models that have a preferred orientation, the orientation they were learned in. And so this is the kind of stuff you do in computer models.
Graphics.
Well, graphics, and also if you do this, if you have a model of a house in the computer and you rotate it, it's everything, anything that has in one model and a orientation of sense to it. So so just complete this picture right now. One of the observations about these cells, these relay cells, is a cell body. They have a bunch of dendrites. They have a lot of synapses on these cells. Maybe 6,000 is a typical number you see for the number of synapses on these cells. And of course the cell has an output axon. I should have drawn this differently. And so, well, if it's a relay cell, why do I need 6,000 synapses? It doesn't make much sense. And worse than that, if you look at these these axons coming from the eye, for example, for a relay cell, you'd expect this axon to go into one cell and that cell to project to the cortex. But there's a lot more synapses here than there are axons coming in. And so these cells, these axons, the numbers are kind of hard to come by, but they clearly connect in far more than one relay cell. So an individual axon coming in is going onto multiple relay cells, which doesn't make sense for a relay, right? Because it's supposed to relay something, it's not supposed to be integrating anything. And so our original hypothesis was that, and these synapses, but almost no synapses go in the cell body. They're always out on the dendrites. our hypothesis was that if a cell has different, if it gets it forms synapses with different axons from the retina, then it would select which one of these is going to relay. It's not going to integrate them. It's going to pick one, and therefore it can say, "Oh, it's a multiplexer." You say, "Okay, my output now is from here, and my output's from there." And so you could, if all the relay cells are doing this, you could basically do any kind of transform you want in this situation. You can route an input to a different input to the same output. And now there's a lot of, we speculated this partly based on observed physiological evidence from other researchers, but also largely theoretical. First of all, I knew that the relay cells wouldn't be doing something as simple as relay, but they look like relay, so the most obvious thing they're doing is rerouting. And then there was some evidence for that, and now there's a lot of evidence for that. Okay, so now we read it last week. We read a couple of papers that were talking about how on a relay cell they can activate individual dendrites separately. So you can think of this as like a one routing mechanism and so on. And there's a lot we don't know about that, but this is almost certainly happening. And so that's all good and cool. So now if you're ready, I can talk about my questions I have. If you have questions about this, you can ask.
Would it be possible in the model we make to have the column just learn every rotation of the object instead of having a thalamus?
Well, that would be very inefficient.
Yeah.
And then you'd have to figure out which of the models is the right one. And then, so you'd have to have many models. And if you don't actually change anything, you'd have to have models for every orientation you've ever seen it. And you'd still have the problem of knowing which one's the right one at the moment in time. And if you're learning one model, then it wouldn't learn the other models. And so it wouldn't be very good.
No, need a thalamus.
Because you also maybe wouldn't have a great understanding of the commonality between the models. So like you kind of think of them almost as like separate things.
Right. So I highly unlikely that's happened. I'm pretty sure it's not. Okay, so this now brings up the question I wanted to ask is, there's two questions I want to ask is, how is the transform learned? Like, how does it know? My assumption this transformation is learned. I'll give you the arguments for it. How would these cells learn what to do this? Assuming that they're not genetically predisposed to do this. I mean, they're genetically set up, but my assumption almost everything in the brain is learned. And I assume that this has to be learned. So that's my first question. And I'll talk about that. And then the second question is, I talked about there's these two different projections back to the thalamus. These are two separate cell populations, often referred to 6A and 6B. In our model, 6A is a sort of a location signal, and 6B is the orientation signal. So the orientation signal makes a lot of sense. This guy is telling this one what orientation the sensor is right now to the model. And so it says, go right now, you're at one orientation, so make this change. But we don't, and yet there's all these projections from 6A, which is location, which we actually no longer have an idea what it's doing here. And I'll talk about that. We don't know why the thalamus needs to know the location on the object. So there's some questions about that. So let me just first talk about how the transformer is learned. We'll run out what I was thinking and see if you guys have any thoughts about it. So imagine you have on the retina, again, this applies to this could apply to your skin too, it doesn't make a difference. The retina, this is the one that's most studied. You might think of a patch of retina as a bunch of these, they're called ganglion cells, the neurons in the retina that project out are ganglion cells. The retina does a lot of processing, but you can ignore that for the moment and just say, okay, the output of the retina is a sort of topographic map of ganglion cells. And I'm looking at a little patch here, right? There's maybe 1 million ganglion cells in your retina, on a retina. So there's a little patch of retina. These are all said that the outputs are coming in and projecting. Now they're going to go. They're going to go to the thalamus, and after the thalamus processes it, they're going to go to the cortex. And the general idea is that there's a topographic representation in the cortex that's similar to the topographic representation of the retina. It's not exactly the same. You don't see cells here that fire exactly like this. By the time you get here, these cells have a more sophisticated response than these. But there's still a topography that's maintained. It's not like it's all random. So this is your cortex, maybe V1. And so this guy is, I won't go into it, but it learns a model of whatever you're looking at. Sensory motor model, like you're moving all around and it learns a model of it. And so, what we need to do is, you can think of it this way, you can think of like, well, if there's no orientation transform, you might just say, well, this cell goes to, to this part, and this cell goes over here, and this cell goes to here, something like that. But, and in the thalamus, you have these relay cells. I'm just gonna draw it like this. It's not really like this, but you can imagine sort of like this. And you can say, oh, there's 1 to 1 to 1 and 1 to 1 to 1. But if I now want to rotate it, do some orientation transform. Well, this cell now has to go this cell has to activate a different one here. Right?
And it's maybe worth mentioning that these connections from layer 6 to the thalamus are modulatory connections, while the inputs from the retina to the thalamus are the driving input.
Right. So that's the point. These don't make the cell fire.
Yeah.
But the evidence is that they do select which dendrite is active.
Yeah.
That's one of the things they do. So they don't, these inputs don't make the thalamus. The thalamus is relaying information, that is clear. But the goal, these things are not creating new information. They're just saying, okay, do something to them. And we think it's an orientation transform. So, so you might think of like, well, imagine this whole thing was rotated, then, this cell here, maybe you can imagine all, these cells have to now respond to sort of a different set. Instead of here, this cell here might, instead of getting input from this, it might get from here or here or here as I rotate it, something like that. I don't think this is I think it has to be learned. And so, cell doesn't really know, it's really stupid. There's probably not enough genetic code to determine all this. We do know that people learn new orientations, they learn new transformations, so it has to be learned. And so the question I was asking, like, well, how would these cells learn to know which place to map over here, right? How do they know that? And that's pretty hard to do. So here's my thinking on it so far. Imagine you start with like a child or a person, and you learn an object first in a canonical form. You just assume that I'm just like I'm learning a word or a letter. It's vertical or whatever it is. And that's the easy way to learn it. You don't show a child letters all rotated. You show a child letters. Okay, here it is, the A, here's B, so on. So you've learned. Let's say you've learned a model, and let's say, for example, there is a mapping from here to here to here. Just say that it exists, but I haven't learned any transformations yet. Now I now rotate this. How does it, and when you rotate it, we can also assume that the cortex knows that it's rotated, and so it changes the signal back to here. But it doesn't know what to do with that signal. These cells this is an input, and now the input's changed. How would these cells learn what to remap to? Is the question. And it's a challenging question. The only way I can think of doing this, and maybe someone can think of something better, is where did I write this down? Here. Imagine you start with, you're looking at, imagine you're going to use continuity in time or hysteresis or something. Imagine you're looking at a simple object you've learned, and now you rotate your head a little bit. And so now the inputs are changed coming in here. Well, if you can make the following assumptions, if you could say, one, I know, perhaps I'm being the cortex, not you personally, you wouldn't know this, it's just the neurons speaking. I know that orientation has changed. I'm going to assume, if this happens gradually, I'm going to assume that the object I'm observing has not changed. So imagine you're a child, you're looking at something, and then you're like this, and then just the default assumption was that changing orientation is not going to change the object. And so what these cells are going to stay active, and they're basically saying, well, imagine I'm getting an input from this guy's getting input from here. And if you gradually change this, well, the input in the retina is no longer driving this thing, but you might think like, well, this cell still has to be active because that's the one that's corresponding to my model. So this guy could say, well, I'm gonna pick, you know, I'm now gonna get input something nearby adjacent to the one that was getting. So the ganglion cell that was active was there, and now I've rotated, now this one seems to be active and this one's not active. I could just say, well, that's the new input I should have based on a gradual change in orientation. I should bring, it was there, now under this orientation, I should bring that one over. And then under this orientation, I can bring that one over. So this is the only way I can think of doing this is that if you don't know anything else, the neurons don't know anything else about the world, if you take a canonical representation and you slowly change it, and then you make the assumption that the model's the same and these guys still should be active, you can say it's like a hysteresis is saying I should still be active because it's the same object. I haven't changed anything else. Then you can say, well, you know, this input's no longer active. Here's another active. I want to put it, and I want to map myself to that based on this orientation. That's where I'm at on this problem.
And yeah, I'm just trying to think because I guess we want it in a way to be kind of specific or sensitive to the orientation that's being sent down and sort of predicted.
Right, but you can
It's
also
like I guess we don't want the cell in the thalamus to activate to that set of neurons regardless of how they are on the retina, right? Because then I feel like then it would.
right. So
it would
what
just
what
be
you're doing,
sensitive to the orientation.
what you would do if you looked at one of these dendrites, you would say, here's let's say here's the big synapse coming from the retina, and then you have a bunch of synapses that recognize the orientation. And so you would say, okay, I'm now going to form a synapse to the retinal ganglion cell, and I'm going to recognize the orientation pattern at the same time. Therefore, I associate that orientation with that retinal cell.
Yeah.
Right. And these retinal cells, they form these really very large, unusual synapses. That literally one synapse would make the cell fire. So that's unusual. That's not like a typical neuron cell. You don't need to recognize the pattern on the retina. You just need to form a big amounts of synapse with that particular ganglion cell. So I mean, this is a bit sketchy because it assumes a lot of things to make this work. But I couldn't think of any other way of doing it. At least I have a way of doing it, but I couldn't think of any other way of doing it.
I mean, so we want it to be a little bit flexible, I agree, but what's the argument against some of it being already kind of hardwired at the beginning? Because it seems like just for orientation transforms, it's not that much to hardwire, and it's like to actually learn it, like for a baby to learn this is super difficult because they can't even control their motor movements enough yet to actually willingly
But
turn the object.
they don't have, they just look at something and their head's bobbling around, that's sufficient.
Yeah, but like things are also just moving.
Well, right, that wouldn't work then. If things are changing all the time, then they'll be confused. This basically assumes that you're fixated on an object that's new, and again, you only have to do this when you're young, right? Once you've learned these orientation transforms, they're gonna be good for a long time. Well, what are the arguments here? There's a lot of arguments for why it has to be learned. We have the inverted glasses argument.
Yeah.
We have the fact that very, very few things in the cortical system, the individual synapses are genetically determined. They're sort of determined to go in a certain place, but the evidence that there are deterministic synapses it doesn't exist as far as I know. Other simple organisms, that's true. If you look at a snail, the snail synapse is completely, the plicia is completely specified by its genome. It's got 400 synapses and they're specified. Every plicia has the same.
But the inverted glasses argument could just be explained by the cortex learning to send different orientation transforms to the thalamus.
That's a little hard because
I mean, it does feel like, yeah, if we could do it just naturally, like, why can't we do it for such an extreme transform? Like, what's special about, and I think in general, like a lot of these studies we found, which were like less extreme than that, it was like something like 60 degrees. As you turn your head, then it becomes very noisy. Whereas like less than 60 degrees, it's quite good. So
Yeah.
it seems like it could be like experience dependent, although it could also be evolution's like, you don't really need to deal with being like upside down that often.
right.
Yeah.
But
I think there's all kinds of just throughout the cortex and there's all these issues of plasticity, where, you know, you can reroute to that column, you can bring in, this is the old ferret experiments where they take the optic nerve and that's going to the cortex and they take the animal at birth and they route the somatosensory cortex to that V1 and it seems to work. This is and then also, think about all the variations of things you might do with somatosensory touch, the different orientations and people have limited movements or some people, it's just, there's so much variability that you could be right, but over the years, I've come to accept that there's so much variability in the cortical system that, you know, you just got to assume things are learned. That's the basic assumption because there's so many examples of plasticity, and I bet you if you did some experiments with teaching people to read vertically, they'd get better at it.
Yeah. I mean, I'm just trying to think of alternative explanations, and I feel like orientation transforms are very, very general. So you could just have this general set of orientation transforms that you apply to any sensory modality.
Right.
And then the part that's actually learned and has all the plasticities in the cortex.
Why would the cortex have to learn anything? If you hardwire it, why would you have to learn anything?
They would just learn which orientation transforms to send to the thalamus.
All right, but
So
that could be
if you have
hard-coded
the
too,
inverted
right?
classes, it would just learn to send the opposite.
But you
orientation.
could, well, you could assume everything's hard-coded. I don't see why you have to learn anything. Well, you know, maybe that's the way to do it for Monty and the Thousand Brains Project. That may be the best way of doing it for you guys. But you know, so I'm not saying we have to do this at all, as I started off by saying, but I think I need some really strong evidence to suggest, there's a lot of evidence suggesting this is learned. Also, there's a limit to how much, you know, think about it, there's a limit to how much each cell can do in this case. An individual relay cell may have, you know, there's a limited number of dendrite segments that it could do this with, and there's a limited number of, you know, there may be 10 inputs from the retina that it can change, and so the amount of the amount of remapping you can do is limited in a neuron, and so the interesting question about that too is it says, okay, I should learn the ones I observe most commonly, and the ones I don't observe very commonly, I'm just not going to be able to do it. It's just too beyond my ability. Right? I can't remap. I can't take a 1,000 axons and remap all 1,000 axons. Individual cells can't do that. It's just not enough dendrites to do that.
Yeah, I feel like, I guess one thing about this is it feels like then each cortex needs to learn a lot of, each cortical column needs to learn a lot of transformations. I'm just wondering, so one thing we've talked about before is
each relay cell has to learn them, right? The cortex is
right, but
it's just-
I guess
in my case, the
fair enough.
learning is
Yeah,
occurring
on
here.
each relay.
Yeah.
But I guess we've sometimes talked about phase codes, and I'm just thinking like a phase code feels like something where that could be a bit more general in that like you don't necessarily need to learn every single orientation, but if you can just like shift it as necessary, and that's doing the transformation.
But
I
mean, so
and then I'm just
so
thinking if
like
relay
you're saying,
cells,
the output of a relay cell doesn't have to remap, it just has to put a different phase on these different inputs.
yeah, and if it's one spike in, one spike out, maybe that's just like changing the timing almost.
Well, the evidence, I don't think they show that. I'm trying to think.
Yeah, I guess it depends also, what it's
Well,
relative
okay,
to.
there is,
but-
when people try to figure out what relay cells are doing, and they typically do this in a slice preparation because the thalamus is in the middle of the brain and they can't really get at it. So they do a dead animal, they slice the brain tissue, so you're really dealing with, a non-realistic thing. But they get a little slice of cortex and a little slice of thalamus, and they see what happens, right? And they activate these cells and see what happens down here. Those studies tend to show some small changes in the output of the thalamus. One of those changes is a temporal shift, a little bit of a temporal shift. Like under certain conditions, they can get the spike to move, a little bit, or maybe it's a little bit broader response or narrow response. So it could be something like that.
Yeah.
That's an interesting idea that a phase somehow a phase code, but it does still seem, you still seem there's a lot of this evidence, you still get one spike in and one spike out. It's not like, oh, you get one spike in a lot of cells, the same spike out, and there are different phases. It doesn't look like that, but again, these studies are so limited, you can't really, it's like, it's hard to know what's really going on because the preparations are so, right?
Cause yeah, for what it's worth, I feel like both, one spike in, one spike out, and also just how powerful the thalamus is when it arrives at the cortex, those spikes fit sort of with a more temporal code. Like if there is a temporal code in the brain, then it's much easier to do if you have like individual spikes have a lot of weight rather than
Right.
like I think of
And
like
then, and there is some, well, there's some really good, there's some other evidence which suggests that too. Let's think about it a little bit. The cortex has to do with a bunch of transformations, right? We've talked about orientation, but we've also talked about scale. It has to do with scale. And the way we think scale is likely implemented is a change in the beta frequency. Theta frequency, I can't remember, it's theta frequency. And so, maybe something like that. Well, I don't want to spend too much time on this because I think in the end, Monty doesn't have to do it this way, right? You can do it whatever you want. But if you're going to do it with neurons, or you're going to do it with like SDR or something like that, you have to think a little bit about this, right? It's easier to do with numbers. You just put a bunch of, Cartesian coordinates and rotate them.
Yeah, I guess the one relevant area for this would be if we learn to represent different spaces than 2D and 3D.
Yeah.
Then you would have to learn
Right.
orientation transforms in those new spaces.
Right.
Yeah, the question is how relevant.
That's another big topic we'll probably avoid today, the 2D, 3D or space thing, because it's really complicated.
Yeah.
Anyway, I just threw this out because, we have a paper that sometime will get finished where, the head-working paper, where we're talking about this concept. And we ought to be at least be able to logically explain to someone how it might work. Because it could be a question a sophisticated reviewer would ask, well, how's this learned? And so I was asking the question myself, like, well, how would it be learned? How would I know this stuff? But I'd rather leave that for a slide for now and go on to the next question, which is even more relevant to the Thousand Brain Project, which is, we got these two projections going back to the thalamus, 6A and 6B. So now we draw the whole thalamus, like a blob of the thalamus, and let's say we'll just call this the part of the thalamus, which like LGM, which is from the retina. So it's a whole bunch of these cells. It's this one here. And then as for those who are around here for a while, we know there's a thing called the thalamic reticular nucleus, and then if you have the cortex here, and you've got 6A and 6B. By the way, there's a lot more cells in 6 than just 6A and 6B, so this is just a subset of the cells in 6, layer 6. We know that 6B, which we think is orientation, just goes in and projects directly to the relay cell dendrites. So that's just what I was talking about. 6A, which we think is location information, projects more complicated. It projects both to the TRN and it projects to the individual cells. And the TRN does some mystery stuff.
And I can't remember, do we know kind of that it's the same cells or it's like it's generally the same nucleus or
well, that was a recent paper we read. Or I read, I don't know if we talked about it here. I mentioned it, where classically, if you read about this, oh yeah, the layer 6A cell projects to here and here, and the layer 6B cell projects right to there. But then there was this recent paper that said, oh, that's not true. They're saying, well, they were tracing this and they said, well, that's true, but what it projects to is a different relay cell. So in the same column, it wouldn't be projecting to the same cells. The layer 6A cells were projecting to here and to here, and they were suggesting, I forget the details of this, but there's a sort of inhibitory surround about this. So what's happening in this scenario, if this column is active, that this cell is active, but this one is being inhibited. The surrounding ones are being inhibited some distance, not just like, it's not the next adjacent cell,
Yeah.
but some distance away. And so that was like, oh, well, that's interesting, because if you look at individual cells from the cortex down to here, they look just like I drew it before. But you have to make sure you're tracing them from the same spot and seeing what the effect they have, and so, oh, it's not the same cells, it's different ones. And so the idea was that recent paper, and there was only one paper, was that maybe it's providing some sort of an inhibitory surround, and then I said, oh, that would be a way of dealing with capacity issues, because a single cortical column may not have enough capacity to do what we want to do. But if I could say on any moment in time, I could say, if I think about cortical columns in the cortex, and here's one column and here's a bunch of columns around it, there's a long been discussion that perhaps what an active column will inhibit neighboring columns, in which case then you'd have a lot higher capacity. You could say, oh, at any point in time on a particular object, I'm going to be using one set of columns, and then another time another set of columns. And so there's some suggestions about that. Again, in the Thousand Brains project, we may not have the limits of capacity that you have in the cortex. Cortex cells have limited ability, that's quite large, but it's still limited. So anyway, so that was a complication there, that these layer 6A cells may not be the same ones that project right to the same cell as the layer 6B cells. But the question I'm having is, why am I sending down a location signal at all to the thalamus? What theoretical possible reason could there be to telling the thalamus the location on the object you're recognizing? And bear in mind, in our current thinking, this is a unique location, so it's a unique location on a unique object. So there's a gazillion of these encodings, SDRs here. And if you're expecting the thalamus to learn every location on every object, that's asking a lot of it. It's possible, but it seems like, well, that's really hard.
Can I
So
ask a
yeah.
question? What's the relative number of neurons in the thalamus to the cortex? Like, what's the kind of
Oh,
magnitude?
well, the number of relay cells is quite small compared to a cortical column.
Right.
It's very small.
Okay.
In fact,
The whole thalamus is, but yeah, I don't know about
like the
neuron
size of a
density.
small, on a human, it's the size of like a small bird egg, you know?
Mhm.
But one thing to think about it is, these relay cells project up to layer 4 in a column. And there's a lot of cells in layer 4, but they only form like less than 10% of the synapses on those cells. And general rule is there's 10 times more fibers coming back than going forward. So these projections, especially layer 6A, there's 10 times more fibers going back to this guy than there are going forward. Again, consistent with the idea that you're not really encoding a sparse coding here. It's not really a distributed code coming up. It's more like this non-distributed code. It's a relay code. It's like a one-to-one code, and you're just rerouting it. That would make sense. That explains these low numbers. So there's relatively few of these. So if there's 1 million fibers coming in from the retina, there might be 1 million relay cells at this point in time. But that goes to all of V1, and V1 has many billions of cells in it. It's just very large. So it's a small number. So, but this number coming back would really, the orientation signal, by the way, is the way we think about orientation in brains. It doesn't take a lot of cells to represent orientation. It's not a sparse code. It's more like a compass direction. You don't take a lot of cells to represent the compass direction. But location takes a lot of cells because we're using sparse distributed representations, and it takes a lot of cells. They're very sparse. So this would be less sparse. This would be very sparse in our theories. So you don't see many layer 6 cells projecting back, and that's consistent with orientation. But you do see a lot of layer 6A cells. In fact, for a long time, people kind of ignored the distinction here because they saw all these cells from layer 6A. They said, well, that's the main input to the thalamus. And these were much fewer of them. They didn't pay much attention to them. But it makes sense from an orientation, and that makes sense for a location. And also, the orientation signals would, in theory, have more direct influence because there's not many active cells. But here we're looking at a sparse code, and therefore the individual cells coming back would seem to do nothing and very little. But it's the code that counts. So
Yeah, I guess one thing we talked about at one point was like, if you had a second column with like a hierarchical, with a hierarchical relation to this column, that like this one, this 6B would, I guess, be projecting to wherever its input is, to make sure that it's, its direct retinal input is oriented correctly. And then there's some kind of relative orientation learned when you have a compositional object between this column and this column, like the output of this column and so on. And that, yeah, maybe that's stored in an L6B connection.
Right.
But I'm just thinking if this projection goes to a different neuron, maybe that fits with the possibility that's kind of going to this neuron somehow, or like influencing what goes into.
You're saying they're just segregated or something like that?
Maybe.
No, with this, I showed it segregated here, so
Yeah.
I'm losing this. Remember, 6B goes,
I should go through.
6A only goes back, 6A only goes back to the retinal ganglion, the thalamic relay cells that project to that column. So this projection only goes to the relay cells that project back up here. 6B projects both to this thalamic nuclei and the next thalamic nuclei. It's black. Remember, like there'd be a higher order thalamic nuclei here, which is 6A is projecting. Actually, 6A wasn't projected down here. 6A, here only projects to this guy, but 6B projects to both. Remember that? That was, and so that's true here too. This big data projects here. So it's complicated. So Vivian and I worked out a hypothesis about how it's really calculating the relative orientation, which is what you need to learn, because the object moves and the component objects rotate even further. So just being complete here, the orientation projects to the input thalamic region and to the next higher order thalamic region, where the layer 6A only projects back to the input region. That's very clear distinction. So this location, so it's complicated, but the orientation, if we want to calculate relative orientation and so on, that all kind of makes sense. But the location's only going back to here. So it's like somehow this guy's telling this thalamic input, here's where I am. Now, complicating this is we know that 6A is a location. We don't know that the exact same cells that represent location are the exact same cells that project down here. There's lots of cells up in the layer 6 that are intermixed with each other. So although it's very clear that there's a whole bunch of layer 6A cells project back to the thalamus, it's not clear that the cells we're relying on for the thalamus are the same ones, although that's what's been reported. So here, the way it looks here is, they say, if you like Sherman and Guillory will say, this cell in layer 6A that projects back to the thalamus also projects up to layer 4. And that is our model, right? And so it's saying, okay, location associated with sensory input. That's our basic model, in which case that is location. If they're right that this is the same cell projects to layer 4 that projects to layer back to the thalamus, is what they claim, that would suggest that this really is the location signal, and it's being projected back to the thalamus.
Or at least it's associated with whatever is in the thalamus.
Well,
in the same way that
but
obviously this isn't, it's receiving location information, but it isn't itself location information up here.
no,
but like this is representing
right.
different, so this might be,
It doesn't mean there's location on it,
but it needs to be.
it's somehow
subjective
this right.
location.
It's like somehow I'm going to learn something about the location that's useful for me here.
Right.
And it could be different. I've speculated that we've talked recently a little bit of speculation that maybe these cells are very phase dependent. And so remember we talked about a set of cells that all the same movement orientation and they have different phases, and therefore the phase determines what the location, but maybe you ignore the phase when you come down here. So then if I have those cells, this is really deep, sorry guys. If I have those cells that are basically all representing movement in the same direction, but there are different phases. well, the cells that are in phase with the theta cycle would be the location. But if I ignore the phase, then all of them are active at the same time. And then if I ignore the phase, then basically I'm just representing a movement direction. But why would I even know that here? I don't know.
Do we know what the TRN is doing as opposed to the LGN?
No, it's inhibitory, meaning the TRN cells themselves project back to the relay cells and inhibit them. We also know that there's a complex sort of network up here. I believe some of these neurons have what they call dendrogenic connections, meaning that they tie together through their dendrites, so they don't act like typical neurons, they more act like a mesh. And it's believed that they're involved in rhythmic generation, like generating different rhythmic signals. So no one knows what they do, I mean, what the function is. But there's attributes like, oh yeah, they're generally inhibitory to these cells. And they spread and they rhythmic and they don't act like regular neurons.
Yeah, I
So
just
you don't
put up
project
a,
back, you don't project back to the cortical columns.
No, these TRN neurons they project to each other and they project back to this neuron. Does that show it?
yeah,
there?
this is I find this one interesting or useful to think through like what you mentioned earlier, how it might be connected to the LGN neurons. So they put like two different ways it could be. And I'm not
this is
sure if
this is the one
there's,
that's showing the inhibitory.
yeah,
Yeah.
so like the classical view was like the A, where the layer 6 neuron goes, and this is excitatory, and it goes to the TRN neuron and to the thalamus relay neuron. And then the TRN sends an inhibitory connection to the same one in the thalamus. Whereas the alternative would be the layer 6 neuron sends excitatory connections to the thalamus and to TRN neurons, but they are different like populations. So basically, these that get input from the cortex then send inhibitory connections to the surrounding relay cells.
That's the idea that you're doing this column inhibition, like
Yeah.
you're just selecting not only, you're selecting the orientation transform for that one cell, but inhibiting other cells.
Yeah.
Which I guess you were talking about capacity earlier, and maybe this is what he meant, is just like, yeah, and also we were talking about before, like the thalamus is pretty small. Like there's only so much information that can come through. And so like kind of attending to what is important
Well,
by kind of like saying, okay, we're only gonna
it was,
process certain,
when I was talking about capacity, I was talking more about the capacity of a cortical column
okay,
to learn, how many models can it learn, like how many location sensory pairs can it learn, like layer 6A to layer 4 type of things. And how many classifications can it learn of that? And we did some early modeling years ago and under a whole bunch of assumptions. So it's, it's for what it's worth. And we found, oh, look, an individual column with a reasonable number of neurons could learn, let's say 500 objects of 40 sensory pairs, something like that, But that's not enough really. And, if we think, so the V1 can learn a lot of stuff. It can't learn everything. That's one of the rules. Even though it can learn complete objects, it can't learn all complete objects, but it can learn some set of complete objects. But it was still a question is, do we need more capacity? So I was thinking more of like the capacity of the cortex itself. If we assume that every column is learning the same thing, well, then it's not as efficient, not as high capacity. If you learn that columns in general, there's a set of columns that are active on, and then a moment later, a different set of columns are active on. So then you're distributing models on different models are put in different sets of columns.
yeah, so maybe it's almost more like that's kind of competition in the long term to make sure that like columns kind of learn, different objects, but then I guess the other is like competition in like a moment in time, which is that like only certain populations of relay cells are active, so that it's not this like cacophony.
I guess I didn't think of it that way. I didn't think that the relay cells would have to compete with each other. If they're doing what we think they're doing, just as, we're not asking too much of them. All we're doing is asking them to remap the relay cell, the relay cell to remap which input it's driving it. That's all we're really asking it. In general, I think of the thalamus as it doesn't know anything about models. it doesn't, it's just like, it's just doing this transformation of input, either sensory input or motor input, it's just rotating, and it knows nothing about the models in the cortex. And so 6A makes sense, 6B makes sense, 6A doesn't make sense, because 6A is model specific. If we're right about it, it's model specific, therefore you're telling something specific about a specific model, and I wouldn't expect the thalamus to know anything about a specific model. I think the thalamus is just a routing mechanism. The model should be up in the cortex, not in this little thing called the thalamus. So that's my problem. We're sending down, if in the way we think about this location signal, that it's unique to objects, we're sending it down to the thalamus. So if the thalamus is going to associate something, it's going to be associating something unique to a particular object. And that doesn't make sense in my mind.
I guess maybe since you said like there are other neurons in layers A, like could be a different subpopulation in that area.
Right, right.
Yeah, like one other figure that never made I'm not sure yet how to make sense of it, is this one which divides it into the parvoconio and magnocellular pathway. And
Yeah.
maybe that's an interesting to look at now in that context as well. Like basically saying, okay, upper layer 6 is the parvocellular pathway and that it basically has the separation in the LGN as well, how like forward and in the backward mapping from layer 6.
I'm looking at this figure, see if it makes sense to my understanding. It doesn't quite make sense.
Yeah.
Well, the problem we have here is that this is looking at primate V1, which has the striate layer 4, which is unique. It's got 4A, B, alpha, C alpha, and C beta. Most of the cortex doesn't have that. Most of the cortex has layer 4, and then many mammals don't have a striate, like a dog and a cat. Dogs, I don't think have a striate V1. Some mammals don't. And so this is like an additional capacity for vision only for certain higher order mammals and no one has any idea why those different layers are there. So this is not your canonical form, right? This is the acanonical, this is the unique system. In my world, the magnocellular cells, the ones are representing, they make sense to represent movement, and the parva cells are representing features, and we can go through why that is again. So I'm looking at this and saying, oh, well, in my sense, anywhere the magnocellular cells project should be representing movement, and they're not showing that here. They're showing only going to 4C alpha, which I don't know what that means. Maybe that's the movement vector. I can't make sense of that. I'd rather see one where it's more the canonical column.
Usually we talk about it going to layer 5.
The border 5 and 6 and the border 3 and 2. Those are the 3 classic inputs, and this doesn't show that.
Yeah, I meant just the magnocellular part we don't really think of it going to layer 4 as much as.
Well, we don't. I hope that this one shows it here, but layer 4C alpha. Some people argue, by the way, against neuroscience is just bizarre. Some people argue that this is a mislabel, that these aren't really different layer 4s, that these are actually different layer 5s and different layer 3s. It's like this is the wrong labeling. You put a 4 in front, and you think they're all doing something similar. No, that's wrong. They say that was this historical accident that they did that. So I just throw that out because we say, oh, it's going to layer 4. Maybe it's not.Maybe that's really part of layer 5, and and who knows what the hell. So I've tried to avoid thinking about these striate meaning layered V1 because then you have to deal with this crazy nomenclature that no one understands. It's worth pointing out though that what makes sense here is the relay cells that are magno and conio and parvo, they're physically aligned to project to the exact same part in the cortex. So the relay cells in here, here, and here all project to the exact same target. They maintain the topology. And so in my mind, if you think of this as motion, and you think that this is like a movement vector, and this is a sense feature, and we can explain why that is, then you would want to implement the same orientation transform on all three of these guys, right? They're all being transformed the same way. Like I'm rotating, my movement has to change, and my sense features have to change. So that makes sense. And you have to keep them separate because one's movement and one's sense features, right? There's some evidence that conio is also movement, but it's often they don't talk about the corneal input coming from the retina, they talk about it coming from the cortical, so I don't know why they show it coming from the retina. I don't know.
What's the corneal one exactly?
It's just another layer. They, you know, they look like little cones, I think. So, but, so there are three basic, there are two big ones, parvo and magno, and then the corneal one is a little smaller one. And also, by the way, the parvo and magno are subdivided by left eye and right eye. So if you look at the classic view of the cellular, the layers in the thalamus, you bring up Google, say, Thalamic layers, you look at an image, you'll see them.
You want me to?
Yeah, sure, why not? People help people. This is, I feel terrible for our new folks here. This is awful introduction to nurse. It's so complicated. Just do Thalamic layers and Google images, you'll see it. Use Thalamic layers? Well, that's the real one. This is the real stain of it, right? LGN, that's the V1. And so you see there's these layers of cell, these all congruent receptor fields. So all these cells have the exact same point on the retina and the same point in the cortex. And then you can see this is left eye, a contra eye, ipsi is the same eye. You know, so these are the parvocellular, there's one eye, the other eye, one eye, the other eye, and magnocellular, same thing, one eye, the other eye. And so they're coming in, there's two retinal, you know, there's two pathways from the retinas, two retinas, and they're being processed all in parallel like that. And then they don't even show the corneal one on here. There is a corneal one, and they're not showing it. It's up here a little bit, it's a small one. So again, this is a classic neuroscience. These guys don't even mention the corneal layer, right? But the other thing, there it was prominent.
Right,
Right?
here it is. It's like in between the.
Oh, it's in between. Oh, I thought it was mostly at the top, and now they're showing the knives. I was wrong about
Or
that.
here it's also kind of.
Oh, there you go. I
spaces
missed that.
in
I thought it
between.
was one place at the top. So maybe that's just different than I recalled. So that's more suggestive that it's doing something different. Just like this layering here suggests that maybe it's modulating or somehow acting on all these at once or something. I don't know. No one really understands the corneal layer, so I don't know what to say about it.
Yeah.
The main ones are the magnocellular, which would be movement, and the parvocellular, which would be features. And why it's divided in two here, no one knows. I've never heard an explanation for that. But it is left eye and right eye. Okay, well, anyway, there you go. That's new for me. I didn't realize that the corneal layers are interspersed between these. So there's so many things we don't know. Well, this is a question that's important for the Thousand Brains Project. It's bothersome to me that there would be a location signal coming down here. Because that means this guy would have to learn unique locations, and it almost can't do that. It's almost like there's no way it can do that. So that makes it bothers me. So like, and then if you believe Sherman and Guillory that these are the exact same cells projected layer 4, you should go back and look at that. Are they certain it's the exact same cells? Did they stain a cell and see one up here and went down here? Maybe. If it's the exact same cells, then it's puzzling to me. And then that's why I came up with this hypothesis that maybe it is sending these signals down here. But if there was phase encoding, then it could be ignoring the phase, and then it would just look like a directional sensitive cell. And the directional sensitive cell would be something this guy could use perhaps, because I'm not asking it to learn specific locations.
I have a question.
Yeah.
So to make associations between two relatively like independently moving sensor patches, like let's say
2 fingers or something like
two
that.
fingers, that has to happen in the thalamus, right?
Why would I have an association between them?
Let's say I re-anchor the grid cells on one of the columns and I want to send that signal to the other, and
Well, that's an assumption that we haven't made yet, right? You start off by thinking like how a single column works. And a single column has to be anchored on its own.
Right.
And if I understand that, I don't have to assume that it's telling another column how to re-anchor. I don't want to start with that assumption, right? You could have 2 different columns. Imagine 2 different fingers, and this finger doesn't really have much to tell this finger, right? is the orientation, this one's rotated independently of this one, and somehow they all work out the same. So I wouldn't make that assumption. I would assume that a single column, I've said this many times, you need to be able to do this. And we need to work out the mechanisms for a single column.
Yeah, I understand.
Yeah.
So, yeah, just throwing out another thought on the layer 6A connection, I guess. So could we maybe think that part of layer 6A is using a shared location space, and then kind of like we talked about last week, in a subpopulation, that shared location space gets sparsified to form a unique location space?
It
And
could.
it only projects the shared location space down to
It
the
could,
thalamus?
that's a lot of assumptions, and that sort of goes in the face. I mean, then you're saying I'm also learning a 6A to 4 based on a shared location space,
Partially, yeah.
partially.
Yeah, like that would be like, okay, we learn the cylinder shape and its rough features, like what curvature is everywhere in a shared location space, but then the specific cylindrical objects like the coffee cup would be like
Yeah.
a sparsified version of that location space.
It's also a little bit inconsistent with the number of fibers coming back from layer 6A to the thalamus. You know, so in V1, you might have 1,000 million relay cells coming in, but you have 10 million layer 6A cells projecting back. Right? That's a lot of cells for a shared space, right?
Yeah,
I mean, it could be, I don't know.
I mean.
What,
depends on how big that space is.
what would the thalamus want to know something about? Why would, the thing I want to ask is, why would I want to tell the thalamus something else? What other information could it use? We need to do two things. It needs to figure out the orientation transform. And we have a sort of a complete solution for that. So we're done. So what other information? It also has to figure out scale. And we haven't talked about scale a lot. Scale, by the way, is when, you have a composite object to people and you say, oh, well, the object could be further away and close. That's different scales. And it could also be relative scale, like the size of this logo to the cup could be a smaller logo or a bigger logo. It has to learn that. And so, we also have to do scale, so it could be related to scale, and maybe scale could be related to specific locations on a specific object. That's something that you could say, oh yeah, on the coffee cup, there's this scale, it has to be here.
Yeah, I guess I don't know if the anatomy is really consistent with this, but I guess that's why I was trying to bring up the relative orientation between a parent object and a child object again, because I agree. This is a really nice solution for and this doesn't change that for calculating this relative orientation. But just in terms of okay, say we have a strong hypothesis we're now or conviction that we're now on this object, this parent object. We move to a location and we want to predict the orientation of the child object and bias that orientation. That's going to be location specific.
Right.
And we need to store that and communicate that some way.
Well,
And that's what I was trying to think.
Yeah. How do
Maybe
we do
this
that
down
again,
connection.
Vivian?
My understanding was it was a direct 6B to 6B connection you guys hypothesized, which it could be.
Yeah, but it has to be on specific locations on the object, right? You have to say for this child object on this location, like I gotta have a coffee cup with a little logo and a big logo.
Yeah.
And a rotated logo and a rotated logo. So I have to be able to learn both of those.
And then, I appreciate the location is a lot of information, but if they really do have such a rich dendritic structure and their main concern is orientation, then maybe that's what they're storing is okay.
Maybe it
I
could
need
be,
to bias this.
right.
And it helps maybe that it's projecting to a different cell, but I don't know if it actually is the right cell.
Another possibility, getting back to this idea of shared location space and non-shared location space. If I look at all across layer 6A, I might have, there's a lot of neurons there, and they could represent a highly sparse, unique location in space. But when I'm projecting down to this relay cell, there's these 10 million fibers coming back, but the assumption would be only a small area of this would project at the small area here, right? It's like there's a topological arrangement. So it might be that they're relying on topology here, the layer 6A aren't converging in the same spot, maybe it's more continuously spread out somehow. I don't know, show this again, that's how did we do this?
Yeah, this is just the proposal for the learning the relative rotation of the child object to the parent object. We didn't talk at all about layer 6A here.
Right, but how do we associate the relative orientation with a specific location on the parent object? That was the trick here. Right, we want to say on a particular location on the parent object.
Well, so we would send the relative orientation of the child to the parent up here to the lower layer 3.
Yeah.
Layer 4 to activate these columns, and then
Right.
whatever feature goes in there would be associated to the location. In
Oh,
layer
right.
6A.
It's done up there, right? That's right. It worked. That worked nicely. I can't remember this. I'm so glad you took this picture. You made this picture. I don't want, again, as with the first question, I don't want to get hung up on this. I mean, I don't want this to get impede Monty and its progression and all this stuff. It is bothersome to me to not understand this, excuse me. It is bothersome to not understand this. I'm gonna send them a message.
I feel like maybe in terms of one potential takeaway for Monty, because like a lot of the transformation stuff that we need to do, we're already doing.
Yeah.
But this idea of like maybe Thalamus is kind of creating a more competitive environment between the columns is something that at some point we could consider.
Right.
Like if we're finding that all the columns are just learning the same objects.
Well, they would unless we did something like that.
Yeah, then
Yeah.
and I mean it doesn't need to be, we don't need a Thalamus, but like just having something that's like.
Right. If we have one of the capacity issues, because some astute observers, when they hear me talk about this, I've had this question maybe twice. Some says, what about the capacity of these columns? How do how many things can they learn? And it's going to be limited. They can't learn everything. So that, we talk about V1 can learn complete objects. Well, they can't learn all complete objects, and it's only going to learn ones that are over small areas of the retina. And so, but if it's still limited enough, then we have a way of sort of expanding capacity. But maybe again, in Monty, we may not have that issue because we can build, unlimited resources into anything we want. But that's a good takeaway. I'm still, ultimately we'll have to do with the scale issue, and we'll have to figure out how scale is represented and how it determines scale per location. It might be related to that. One of the reasons to suggest that is that, remember, if this is a location and the TRN is associated with various oscillations. I don't remember the details, but that you could be saying, I'm selecting a particular oscillatory, scale here. I'm telling it what scale to do based on the location. So maybe this is time orientation, and that we're going to use this for determining scale somehow. I don't know.
Yeah, so I think one thing that's currently different in the Monty implementation to how we talk about it in the brain is that in Monty we do the transformation of taking the current location, adding the movement, and then getting the next location. We do that outside of the cortical column, and then give the column the next location. Whereas here we're saying the column gets the movement vector and does the location transfer.
Yeah.
But is there a way to think of that actually happening in the thalamus? So basically this layer 6A cell tells the thalamus, okay, I'm at this location right now. The thalamus gets the movement vector and does the path integration. I mean, I know that's a bit of a crazy idea, but
it doesn't sound like it doesn't seem like there are enough cells in the thalamus to do path integration.
Yeah.
It's just kind of a simple structure.
Yeah.
There are, okay, so let's be clear. If you look in the thalamus, there are basically two types of cells. There are relay cells, which we've been talking about exclusively. There's the TRN, that's separate, but there's also something called matrix cells. And matrix cells I have always been working for many, many years. I've been working on the assumption that these are the cells that determine timing for sequences and movements and so on. And there's a lot of evidence that accumulated over the years that would be true, that these cells, I think they project to layer one, and they essentially could be providing a timing signal, like if you're trying to learn the durations of elements in a sequence, like the duration of notes in a melody, that these guys would be like a little countdown clock that says, okay, beginning now, you can count about a second, and then you have to reset and go another second and so on. I mention that because there's more, right? There is other cell type in the thalamus. These are very different than relay cells. They don't get, as far as I recall, they don't get any input from the sensors. They're more like they get input from the cortex. It's like the way I think of them is they're told, like, when if you listen to a melody, notes always have like an attack, the first beginning of the note, and there was some evidence that these cells become active at that attack, and then they play out a timing signal, which then gets sent up to the cortex, so the cortex can learn the duration of individual elements and sequences. I mentioned that because beyond those two, I'm not aware of any other cells in the thalamus. There might be some, but I'm not aware of them. So
Yeah.
what were the cells that would do path integration? Oh,
Yeah.
they don't look like they'd be there. By the way, in one of these other papers, did I print that one out? Or it's on my computer? I think it's on my computer. Yes, on my computer, I can show it to you. Where was I gonna go with that? Why did I bring that up?
Well, yeah, I mean, it could be some interaction with the TRN and that one inhibiting some of the cells in LGN, but yeah, totally different mechanism than the grid cells, so.
Right, so I always thought the general rule for the thalamus in my book was always, it's generic, meaning it doesn't know about specific objects. And it can do orientation transforms, it can do scaling, and it can provide a timing signal. And it can modify that timing signal, because the trick here was sometimes you want things to be sped up and slow down, just like you want to be able to scale time. You could scale space, but you're scaling. This is a scaling of time, so you could change that rapidly. This signal, when you want to play back a melody faster than you learned it, these cells would speed up their timing signal. So these are things that apply to all models. It's like a timing mechanism, and a transfer mechanism, and a movement, transformation of movement and scale. So this would be scale for timing. It might work for scale for other things too. But so it's generic. It's nothing specific about any particular modality, any particular models. That's how I do it. So I don't think there's other cells in here that could do like path integration. There doesn't seem to be enough cells.
Yeah, it would have to be something in the TRN.
TRN is pretty weird.
Yeah.
It doesn't look like it would be doing that. The TRN looks like it's some sort of they often refer to as an intentional mechanism, like if something occurred, that you didn't anticipate, it would essentially open the gates to other cells. So, for example, in this example where you have it, well, you don't have it up there anymore, but the one that showed the thalamus where the inhibition.
Yeah, I can open that.
You had it there.
I had it on the other laptop.
Oh. Not that one, that one there. One of the things I did is this, that on B there, you're sort of inhibiting these other relay cells nearby, but when you do, they're in this pre-bursting mode thing. So if an input comes in that's unexpected, those outside green cells would burst. They fire rapidly, and then they reset everything. And so it's like if I'm attending something here and then there's a little flash of light off to the side, I lose my attention. I move over there and attend to that. So it's associated with that, and so the relay cells, the TRN cells, the TRNs are associated with this resetting of attention signal. Like something unexpected occurred, and then you have to attend to something new as opposed to what you were attending before. So it's a lot of literature about that.
That's interesting. So could that be a clue as to what the L6A signal is, like that the location signal somehow encode like if there's a sudden prediction error in the location space.
Right, in fact, that's what a lot of people have speculated in like, in the predictive coding world, that's what's happening, is that the cortex is telling the thalamus what to expect. And if the thalamus doesn't get what it expects, the cells burst and it attends to something else. So what I have a problem with that is, I could imagine the thalamus being told what feature to expect. You know, like, here's a feature, you should be expecting, you know, this line or something like that. But I have trouble explaining how it could say why it'd be specific to an object. Right? It's like, it's just, the specific to the object part is like, it doesn't make sense to me. I don't want to spend too much time on this. We've already spent a lot of time on it. I don't know if we have time for more stuff. This was an interesting paper because, going back a little bit, layer 2, 3 in the cortex are often just lumped together, layer 2/3. And people didn't really know why, you know, and some people talk about, where's my mouse? Okay, up there, yeah, okay.
You want me to go to a figure?
No, well, maybe in a second. Yeah, go to the figure 1. This is a very misleading figure. Let's point out, very misleading. So in our models, we've looked at it like this. We've said, we have essentially layer 6A and layer 4 are bidirectionally associative, connect to each other. So this is a location, and this is a feature, and you're essentially associating features with locations on an object. That's a basic model. But then you need to form a representation. Sometimes you don't actually have to do this, but sometimes you want to put a label on this object, like what is it? Then you'd have another layer, which we've always said is like layer 3. And this is what we call a temporal pooler. Basically, it says, I take all these associative pairs, like these representations represent a location and a feature. And I say, and since it's unique to the object, and every one of these representations is part of this common object. So this would be the object, and this would be the location feature pairs. And so an object is a bunch of location feature pairs. That's it, right? And so this would be like a stable representation while these are changing. So it's like, oh yeah, my finger's moving over the coffee cup, but it's still a coffee cup. I know it's a coffee cup, but my perception is it's stable. And this layer 3 cells project to the next broadly. So, but these cells don't. So basically you're aware of the object you're touching, the object you're sensing, but you're not really aware of, unless you attend to it, unless you make a point of really trying to notice it, you're not really thinking like, where are my fingers on this object and what are they feeling right now? You know, you just pick it up and say, yeah, okay, I'm just holding my coffee cup. I don't say like, oh, my finger is on the little lip of there. You can do that, but generally you don't do that. So basically, okay, I've got this object, and that's our basic model. And these operations have to occur. You have to be able to do this. This is like what inference is. It's figuring out a common representation for a series of changing sensory motor inputs. So, so that's that, and this becomes the input to layer 4 in the next higher region, and there we in the paper we were writing, they're saying, okay, this object now is a feature of the next object up in the hierarchy. All that really looks nice. So it makes me feel good about that. But what about layer 2 and 3? Because they're not one thing. So this paper that we see on the picture up here, what they did, they were trying to measure the physiology of layer 2 and 3. That is, what do the cells respond to? They weren't looking at the anatomy. So that is, they weren't looking at what layers 2 and 3 are connected to, but they're looking only how they respond differently to inputs. And what's misleading about this figure, this is an anatomy figure. And they don't show layer 3 projecting to layer 2. And in the paper, they point out, they say, well, these inputs to layer 2 are not well known, and we didn't show them. They may come from layers 3, 4B, and 5. So basically, if you look at this figure, you would think that layer 3 is not connecting to layer 2, but here they say, yeah, might. We don't know. We're not really paying attention to that. And other people have clearly stated that layer 3 projects to layer 2. So if you just start off with this paper, you get misleading representation of what's going on. This is like classic neuroscientists. Neuroscientists are ignoring a whole bunch of other data, makes this picture which would confuse you. So we don't really want to look at this paper, this figure. This is really misleading. So, and it's the first figure in the paper. So you go down here, keep going. Let's see. Yes, that one's important. So let me just talk about what's going on here. So the upper line is a layer 2 response property, and the bottom line is a layer 3 cells. Okay. And what they have is a monkey is sitting in a chair, its head is fixed, they're displaying something on the screen. The monkey is trained to stare at a dot on the screen. He's thirsty and he doesn't get water if his eyes move from the dot on the screen. So this poor monkey, this is terrible. And then off to the side, not where he's staring, but off to the side, there's a part of the visual field where they're measuring these cells. These cells are responding to that part of the visual field off to the side. And they put a sinusoidal grading in there. They basically put lines moving in a particular direction and then another direction. So here's the big difference here. There's a couple things going on here. These cells in layer 2 and 3, the ones that they're showing results for, are movement sensitive. So the cells respond when there's an oriented line moving. So you can see above it, there's like a long line and a short line, and they show little arrows on it, right? So imagine it's not just a line, but it's a sinusoidal grading that's moving through space. And they're showing how wide the line is, the cell prefers the width of a line. So how wide should that be? So you can see layer 2 prefers longer, much broader things. And layer 3 prefers much shorter lines. But the real big difference here is that in layer 2, the cells respond if the line's moving either direction. So on that top line, there's every half a second, the cell, or maybe every second, I think it's every half a second, the cell, no, every second. Well, every half a second, it changes direction. So it goes half a direction, one direction, half a second, the other direction, half a second, another. And you see the layer 2 cells respond in both directions. So if it's not, when the cell starts moving, it goes, oh yeah, blah, blah, blah. Those little vertical lines are the cell spikes. Okay. So the layer 3 cells respond in both directions. The little thing on the right is just indicating it's a plot showing how they respond in 2 directions. And then the layer 3 cell responds to movement, but only in one direction. So when the lines are moving one way, it responds. The lines are moving the other way, it doesn't respond. So this is one of the primary results of this paper. Also that the layer 2 cells are much less picky. The line can be really big, it can be fuzzy, it's not very precise, where the layer 3 cells seem to be much more precise. Like I need to have this skinny little line at this exact orientation, and it has to move in this direction, and then I'll respond. Okay, so a couple of things come out of this which are very problematic for us. One is, if layer 3 cells are the input to the next region, if he's actually measuring, or whoever they are, scientist is, if they're actually measuring the same layer 3 cells and projecting to layer 4, those cells are only motion sensitive only in one direction. That's not what I just said. I said these would be stable over multiple movements of the object. And I have some thoughts about this in a moment. Then layer 2 also, sometimes there's evidence that layer 2 also, and they showed it, layer 2 also projects here sometimes. So I'd have layer 3 and layer 2 and these different things going on. What the hell's going on here? Why are these motion sensitive at all? We don't want them to be motion sensitive. So before we get all bent out of shape about that, there's another paper I read, which discusses an issue we've talked about a lot, which is the difficulty in figuring out what cells you're looking at and why you're looking at certain cells. So they take the monkey and they show moving sinusoidal grades. They don't actually see what happens if there's an image. They only show movement. So you're only going to find cells that respond to movement. These, they have to look for these cells. They're not easy to find. So they find one, they go, oh, this one's responding. But the vast majority of cells aren't responding to these inputs. So just remember that. So it's like, they don't say that. Well, they do, but it's kind of hidden in the data. So this could be very misleading, right? There could be lots of different cells up here. We've talked about the possibility that you're doing path integration up here, that is, the object is moving, not in space, but it has its own behaviors, like a staple opening, closing. That might be represented up here, and these might be related to path integration in object motion, object behaviors. So it's very misleading. But I still thought it was interesting that they were able to find cells that the distinction between layer 2 and 3 is one cell's responded in one direction, one responded in the other direction. And this gets really obtuse because in the path integration, I'm sorry, for the people, you're just not going to understand any of this. But the path integration technology we've talked about, you really like to have cells that are sort of phase encoding but moving in both directions, not just one direction. So this was suggestive, like, oh, maybe there's a path integration, maybe layer 2 is doing path integration, and it's really representing the location or this path integration of movement of components of an object.
It would encode like the state of the object.
It would call this, well, you'd have to, it's encoding the state, but yeah, it's like, it would be the predecessor to the state of the object. It'd be like saying, okay, the stapler is opening. The stapler is not moving relative to me, but part of the stapler is moving relative to the other part of the stapler. That is different than me moving, that's not the same as a stapler moving through the world or me moving through the world. And so these cells might be doing the path integration of that component. It's just,
the gate of its cage.
with that,
The gate of its cage that it's learned.
the cage.
Yeah, the monkey generally lives in a cage. And sees a door.
Oh, maybe. Well, the monkey doesn't get to see that. The monkey just looks at the screen and looks at the dot all day long. Until it's safe. They say, well, we did this for about 2 hours until the monkey is satiated. So they get the monkey really thirsty, miserable life. And then it gets a little drop, little drop, and it lasts for 2 hours until once he's no longer thirsty, he stops staring at the dot. I just thought it was interesting. We talked about layer 2 and layer 3, and we didn't really have, what's the difference? We talked about classification. Well, there are clearly sets of cells up there that are motion sensitive, and there's a differentiation between layer 2 and layer 3. And I thought that was interesting. I'm filing it away in my head as like, oh, this is a clue to the idea that there might be path integration occurring up there relative to object behaviors. That was, which I've speculated for a long time. That's where it should be. That's the lower input to the lower layer 3, upper layer 4. That would be perhaps motion related. And so there's some evidence. Okay, yeah, it's processing motion, and it's probably going to do something. Although it doesn't make sense to send those. I don't want to send that lower line signal to layer 4. If this is supposed to represent the object, I'm pretty sure it does. Then I'm saying that there's probably different cells in layer 3 are projecting to this layer 4 than these cells.
Sorry, this was discussed, but like when sending that signal, I guess movement itself, I mean, is that kind of the object state that we might want to send to, like, at the object level, rather than like they might be seeing it when they do some sinusoidal gradients or whatever. But if this is more kind of encoding the movement of the whole object.
This would be movement. Well, to me, this would be a movement of part of the object.
Okay, I was just wondering, because if it's movement of the whole object, that might be more useful to the next layer.
Movement of the whole object meaning like through space, or meaning it's changing?
Or like changing,
Okay,
like,
it could be
yeah, like a
both.
behavioral state.
Or then I would give, this is basically any kind of change to the object itself.
Like, yeah, it's closing or whatever.
Yeah.
Whatever, it's
Yeah.
morphing, it's
Yeah.
bird flopping its wings. The point being is that you see that the line down there, on the B1, you see how it's restricted to a small dimension. Basically, if you showed the whole field of vision moving, these cells wouldn't respond. They don't want to
Yeah.
respond to you moving through the world. They want to respond to some part of the world that's moving, and the rest of the world is not moving. So that's telling you it's focusing on the object itself. That's a suggestion of that, and not the whole world. And if you look at the cells and layers, the lower layers, they respond to very broad areas of their vision. So that's more like motion to the world.
And it might not even be doing the path integration itself in layer 2, 3, but rather the path integration could be happening in the lower layers, but then layer 2, 3 would just encode the state of the
Maybe, but-
object,
but-
which
but
is
don't-
changing.
But wouldn't you need to, I don't see how you could do in the lower layers, because, I mean, it's a different, it's not the same I mean, one, on path integration, my location in space, like my body's moving, right? And that has to be maintained separately than the object's moving. So I can't use the same mechanism for those two, right?
Yeah, but you could have, so first, I'm not sure how we would do path integration in layer 2, 3 themselves. But like a way you could do it in the lower layers would be that like each state of the object is a kind of like a different model, but then you learn an association between how to move through the different states of the object.
Yeah, if you think about the stapler, you could argue that the higher region would be looking at an object composed of two components in the lower region, and the relative position and those two components are moving relative to each other. It might take a
Yeah.
hierarchy to do that one.
Yeah, but still, I feel like the relative locations in which those two parts of the stapler can be in would be encoded in the layer 6A to 4 connection.
Maybe.
Or
It's more like it'd be more that's what we were talking about earlier, that we have to learn the relative orientation of two components, right? Two objects. It'd be more like that relative orientation is changing. I have to learn a sequence of relative orientations.
I'm just saying it like, I mean, we don't really want to need grid cells in layer 2, 3 as well. and-
Yeah, but what would I do with these
Representations
cells? Why
up
are they up
there.
there? What are they doing then?
I mean, it would make sense if they just encode the state of the object and that state changes as the object moves. So like, if it encodes the state of the stapler, which is neither completely open nor completely closed, then it fires every time the stapler goes into that.
Yeah, but it wouldn't, but once it stops, they would stop firing, and therefore they wouldn't encode the position. They only encode movement, relative change of that position. These cells would stop firing if the stapler top stopped opening.
Yeah, okay, yeah, that's a good point. I guess it might be part of the state of the object, whether it is currently in motion, but yeah, I don't know.
I don't know either. We're, for the newcomers here, we're bringing up all our dirty laundry, all the things we don't understand. But we understand a lot, trust me. But the research meeting is not to go over what we understand, it's to go over things we don't understand.
Yeah, I'm just trying to figure out a way to explain this without needing like grid cells and location representations in layer 2, 3. as
Well, I guess since these are temporary, these particular cells wouldn't fire if the object stopped moving. Then they seem like they couldn't represent the state of the object. They can only represent the movement of the components of the object.
Well,
That's
there
what-
is no, the monkey never sees the rod stopping, right? It only sees it.
Well, it stops it does stop at the transition points. It reverses direction and cells.
Yeah, but the event will be, or the state of the model will be
Yeah.
in motion always.
Yeah.
Like,
I'm pretty sure, I'm not 100% sure, Rami, but I'm pretty sure, like 98% certain, that these cells would not respond to a static image.
Yeah.
Pretty sure. They would respond to motion only, but I can't be 100% certain about that, but I'm pretty certain about that. So again, there's lots of cells in these places up here, and they pick the ones that they're recording from based on how they set up their experiment. Oh, like your husband, and your baby. Might be important. Anyway, it was more like interesting data. I was like, oh, I never saw this. This is interesting. And I was like, what am I gonna make of that? Who knows? Can't figure that out yet. All right. These are things that I'm gonna file away for some future under future discussion. For the newcomers, there is this concept that we up to now, all we've done with Monty is recognize static objects, the movement sensation, but it doesn't really deal with the issue of objects that are actually moving themselves, have behavior. I call it behaviors, and therefore you have to be able to say, You know, a bird with its wings out is still a bird with its wings in, you know, and if it's moving back and forth, that's a typical bird, but you wouldn't see a bird moving one wing out and back. That's not, somehow you know that, you've learned that, right? So the model of a bird has to include behaviors, or the model of a staple has to include behaviors that typically observed, you know, opening, closing, staples come out, you know, various things like that. And it's a pretty important part of sensory motor learning because so many objects have behaviors and we push and touch and manipulate them. And we don't really have that worked out yet. So we do have like, oh, how does it recognize objects through multisensory touching and seeing and things like that. Okay. I don't think the problem, there's answers to these questions and we're getting close to them, but at the moment, it doesn't, you know, may not seem like that. And then I have one other, unless someone wants to discuss this further. I just thought this was interesting, this paper. Let me see. There was this paper called about concept cells. Is that the one I have here? Yeah. Yeah. Okay, so just, what's this paper about? This paper is about in humans, in their hippocampal complex at the hippocampus and entorhinal cortex, which is called the medial temporal lobe in humans. So they talk about MTL, but that's the same as hippocampus and entorhinal cortex in rats. So you have to know that. So in humans that are awake, because these humans have electrodes planted in their heads because they have intractable epilepsy, and they're going to remove part of the brain to stop their epileptic seizures. And so for like 5 days, they go around with these electrodes in their head so that the doctors can try to figure out where the epileptic seizures are starting, and then they know where to remove tissue. So during those 5 days, the humans agreed to be subjects in experiments. So they have human data. Maybe I should project from this. Can I project easily?
Oh, yeah, sure.
They have human data for this. And they can measure from individual cells or a group of cells at once. So that's what we're talking about. And concept cells is a name for what some people refer to as, a number of years ago, people found doing these experiments, like the subject is in the, got the electrodes in its head, and those electrodes are in the medial temporal lobe, which is deep in the brain, in the entorhinal cortex and thalamus, the hippocampus. And they find cells that respond to concepts, like the famous one was Jennifer Aniston. I guess there's an actress. I don't watch TV, but she's Friends or something like that.
Yeah.
Yes.
Okay, so Friends. And they say, oh, there's a cell that responds to Jennifer Aniston. And not only that, it's not just a particular, it's like anything to do with Jennifer Aniston, like, you spell her name out and that cell responds, or different positions, or, so
Like her name written and stuff.
right. So it's like, oh, well, so these, like, they call these concept cells, right? And so this was about, what did I write about this? In my notes here. Well, no, I guess it's down here. So this is about that. It wasn't particularly interesting. They're showing in a human, this is where the hippocampus is and the entorhinal cortex. But what was interesting about it is like the whole idea of like a concept cell is like a grandmother neuron. Like this neuron just responds to Jennifer Aniston. That doesn't really make sense. You have to have a lot of cells respond to Jennifer Aniston because you can't assume that one cell represents Jennifer Aniston. That's nonsense. No one believes that. And then how many different things can you encode? And this gets to the nature of sparse distributed representations, because we've argued that most of the things that when you represent an object, you're doing with this very sparse code, the individual cells don't really, they'll always respond in the same for that object, but they'll also respond to lots of other objects too. Like, so if it's like a 2% sparsity, that cell should become active every 50 things. And so this paper got into these questions here. I thought there was some interesting piece of the data here. So it says here in a population of 1 billion neurons, less than 1 million are involved in the representation of a given concept, such as Jennifer Aniston or Halle Bailey. And each of these MTL neurons may encode up to a few dozen of the 10,000 to 30,000 things a person can recognize. So they're saying, okay, so maybe these things respond to maybe a dozen different objects. That's not enough, right? That's just not enough. I mean, it doesn't, the math doesn't work out. But then it says, however, both these estimates should be taken as upper limits. The true values may be a couple order of magnitudes lower. Okay, that's a big caveat. A couple order of magnitudes lower. And because it's difficult to select very selective neurons. So let's go down to this box here. They're just talking about if I'm looking at a particular neuron and they're trying to get to see what it responds to, the vast majority of neurons seem silent. They don't respond to anything. And so they just figure they're not doing anything. But in our world, in a sparse distributed representation, they would be, you just didn't get the right stimulus, right? So here they said, they're talking about individual neurons here. And they said, well, there's 3 neurons we're looking at here, these blue one, the red one, the green one. And they say the third neuron fired only, these 2 fired a lot of spikes, 6,700, 1,800 over some period of time. The third neuron fired only 218 spikes during the 30-minute recording. So they're recording for 30 minutes, showing all these images, and they only got 218 spikes out of 30 minutes. That is like noise. That's like so little, right? It's very, very little. And yet they were able to, and then they could show what that cell seemed to respond to. Like, well, respond to these 2 different towers and some rabbits, and so I thought, and these cells responded to 40 times more. So what they were basically arguing in this paper, that the data that you typically get from these experiments, they try to figure out, come up with things that someone would know, like, oh, they watch TV, let's show them Friends characters, or let's show them pictures of their family, or show them pictures of the researchers. These are people they know intimately, and they see if they get cells to respond to it. But it turns out maybe most of the neurons are not, that's really biased data, because most of the neurons are still representing things in the world, but are extremely sparse, and you just wouldn't know it, and you don't see that. So this, Niels and I have had this debate a while about, our neurons, as a group, we've talked about this. But they made some good arguments in this paper why it's very difficult to really get a proper representation. Let's see, here another one's here. I'll just go back up to the top here, right? I was taking notes here. In addition, the images were concepts that were very familiar to the patients. For example, pictures of patients themselves. All right, so they're showing your own picture. Family members, experimenters, and celebrities to increase the probability of getting a response. Indeed, personally relevant items are shown to elicit the largest number of responses. And most of the things, 10 to 30,000 things a person can recognize, may not be represented in the image at all, as these may not be silent enough to trigger memory process. That may not be true. Basically, they also later they argue, well, they might be represented in the MTL, medial temporal lobe, but you're just not going to find them because they only spike every 200 every 30 minutes, spike. Anyway, there was some interesting. If you're interested in this topic, there was some interesting data in here. That's what I thought. It was like, oh, this is interesting data. They talk about the problems of detecting and the bias that they have. When we talk about, oh, they find these cells, they seem to respond to concepts that are very explicit. That's the exception. The vast majority of concepts that things you could do, they wouldn't find, they're not very familiar. And they gave an example, one cell that seems to respire, 218 times over 30 minutes. And it was fine for certain things. And they basically said we were lucky to randomly find something that made that cell fire. Most cells, they said you would find something that would make it so.
So just to clarify, they're arguing basically that they're not claiming that it's a Jennifer Aniston like grandmother cell. It's just like, it's still a distributed code, but the only thing they found that this cell responded to happened to be Jennifer Aniston.
Right, and obviously there's also arguing that
there will be many other things.
Right, but they also argued, and there was a nice figure here. Is it in this paper? I think it is. Yeah, this figure here. So this is interesting. So these are cortical regions. The pink one here is V4, and this is upper auditory cortex, right? So these are places where you already expect objects to be recognized in these 2 cortical regions. And they say the cortex already has representations of Jennifer Aniston or anything else like that. And then the blue ones are the medial temporal lobe. So this is the entoronic cortex, the hippocampus, and these two things parallel. These are like 3 steps in processing. So you're already feeding in, you're not feeding in raw sensory data. You're feeding in representations of Jennifer Aniston already into this thing. So our argument, we've talked in the past, is like the columns themselves, the mini columns, they represent, you might have Jennifer Aniston mini columns type of thing, it's like, because it's so highly represented. Anyway, it's just like, they kind of ignored that. They kind of said like, like you're starting, you only really get Jennifer Aniston further up in the hippocampus, but anyway, it was interesting data. But I thought this was fascinating because our basic theory is that these blue things are the predecessors to a cortical column. These existed perhaps in older animals, and they were just the entoronic cortex and hippocampus. And a cortical column is basically a collection. It is like similar to this. And entoronic cortex is location. Hippocampus is like location cells, like object feature cells. And so they basically, the cortex has its own model of Jennifer Aniston. And then it feeds that into this blue thing, which is basically learning temporary memories about Jennifer Aniston, temporary associations, like, oh, I saw her in this movie yesterday. Anyway, it was really interesting how they talked about why you might not see, why the data is biased, and why they would not see typical representations because the data is so biased. And so it's just a warning that we've talked about, and it was nicely written about in here. I'm not gonna try to read all this for you here. As mentioned before, concept cells do not act in isolation, but as far as cell assemblies. This is what Niels wants here. So here they're saying this is speculation. They're speculating, and this is what you've speculated, Niels, is that I have representations for Luke Skywalker, Yoda, and Darth Vader, but some of the cells are common in those representations. So that when you see Luke Skywalker, you get some of the same representation as Yoda, and so these are all Star Wars things. And the behavior, it kind of acts like it, but they didn't walk through the numbers to see if it actually works, which is the problem I have with this. But they're arguing this might be what's going on, which is what you've argued, Niels, that there's these overlapping bits, but they didn't, in my opinion, they didn't walk through it in detail. They just threw it out there.
And yeah, to be fair, my understanding that's also something you meant to use to.
Yeah,
like,
right. I argued for this. I give talks on this. Yeah, I know.
And I think Sukutai even still.
Does he still do it?
Yeah, because he's like, SDRs should have this kind of.
Yeah, but I just don't know how it works. Maybe I need to spend more time on it. I used to believe it. I don't believe it anymore. Maybe I'm wrong. I don't know. It's difficult to. I can walk again through the difficulties, but here they're making. This is a picture of what you've argued, so it might be interesting to.
Yeah, well, that's a nice drawing of that.
Yeah, sort of. So this paper talks a lot about this stuff, so I thought that was interesting.
Yeah, thanks.
Yeah, that's it. And again, these are papers that I thought were interesting numbers. Just throw them out there because the stuff that's worth keeping track of.
Just
Okay.
like an interesting curiosity. I mean, I don't think it's necessarily that significant, but there was kind of a paper a couple years ago that basically showed similar things in these like transformer networks from like OpenAI and stuff, where they found like a Spider-Man neuron or a Trump neuron that would activate to like Spider-Man written down, the Spider-Man logo, Spider-Man images.
Were those sparse representations or were they?
So I think they kind of argued something similar, that it's not like this neuron is only active for this, but yeah, they were relatively sparse, I think.
Well, I mean, because the problem I have with this coding like this is when you get very sparse. That's the problem. There aren't enough neurons to do the overlap. Right?
Yeah, I mean, I guess those are real value neurons, so maybe they have higher representation.
They're all kind of the real value neurons, and they have very specific real value synapses, and they're not like sparse. You're not really doing a sparse code, right? Or are you? I don't know. Seems like real value neurons, you're not relying on the sparse code.
Yeah, this was a regular clip. They show a model, an image, and then they show the text of that image, like captions of that image,
Yeah,
and then they map those two representations together in dense space. And then so they're real valued and they're dense, they're not really sparse coded.
Mm.
But then they learn this mapping, and then they look at the individual neurons, what output they activate at the end. Or actually they go backwards, they go from an image, they look at the representation, then they go backwards and see what neurons are responsible for activating that image. And then they find, oh, this neuron is responsible for all of the same concepts, kind of like.
yeah. Well, we've talked about, I wrote this down, I wrote it down someplace, yeah. There's different ways neurons can represent stuff. And this may or may not be useful. I don't really write it down. Is it okay? We don't. That's something I can do.
I think it would be worth it sometime to have an explicit session just talking through this idea of SDRC coding meaning and
Right.
come to a conclusion if this is something we should keep pursuing or
Yeah,
not.
good idea.
There's a lot of that in HTM school as well. So we don't
Yeah, I
need
know,
that.
I know. It sounds so great. I just,
Yeah.
so I thought, we're trying to one of the big questions you have is how do neurons represent information? And the classic way going back a long way ago is you start off with firing rate. Meaning the neuron spiking, and the faster it spikes, it's a graded signal, right? And there are lots of examples of that, like a neuron that's innervating a muscle fiber is basically a rate coded neuron. Basically, if it fires faster, the muscle pulls tighter, and if it fires less, so. And there's lots of neurons in the old part of the brain releasing chemicals and so on that are firing rate. But firing rate, it's hard. Well, that's one thing, right? But we don't believe that's happening everywhere, right? Another thing you could do is you could say, well, an alternate is you have a sparse distributed representation where so you might have 1 million cells and 2% are active. So, you have 10,000 cells active. That's a lot. Could be 10,000 and 100. But the point here is that the actual rate of the firing makes no difference. It's which cells are active at the same time. So this has a very high capacity, meaning you can represent a ton of stuff. Capacity, unlimited number of representations, and it has all these other desirable attributes which Subotai wrote about in our neuron paper. The noise tolerance, robustness across, it's amazing. It's an incredible, and we know that in the brain, or in the cortex specifically, there are lots of sparse representations. So we're pretty sure that's happening, and it's happening in the cortex for certain, but it doesn't mean everything is in the cortex is working that way. We also have another idea, which is there are the mini columns in the cortex, which is this would be like you might have 10 mini columns representing different orientations, and what you have is 3 active at once. And so you're just representing a 360-degree orientation, and you're moving it, it's like a 3 of 8 type or 3 of 10 type of representation. Not high capacity, it's not unique, but it's simple, and you can do path integration on it, and it suffices for many things. And so our basic neuron theory combines this plus this to form sparse representations. You start with a non-sparse mini-column representation, and you sparsify it by picking individual cells, and it has all these little good properties. Another one that we've learned recently is representation that was before. This is the phase, using phase. So this is the idea that you see in place cells and grid cells, where you kind of have a representation which is, it's sort of in between sparsity. It's not super sparse, like the grid cells and place cells are not particularly, they're sparse, but not like super sparse. So you might say semi-sparse representation, but the same neurons, different cells fire different phases. So if you're looking at grid cells in a single theta phase, meaning like I don't know, it would be 100 milliseconds, something like that. You go through a series of representations in order. So you're packing in a bunch of them right in order. So you might have, representation 1, and then representation 2, and representation 3, and these occur very rapidly because it's the same neurons, but which ones are in phase with the theta cycle changes at any moment in time. So you have a series of representations in time, and this is clearly happening in parts of the brain. And so this is like, oh my gosh, could this be happening everywhere in the brain? Could you be, in addition to SDRs, could you also be doing phase versions of SDRs? That's essentially what this is doing here.
So the decision of which cells are attached to which phase, is that hardwired encoded in the genetics?
Well, I don't know. I don't know if anyone knows that question. This comes about from studying grid cells and place cells.
Right.
And they see this phase procession. This for the new people here, an animal is moving in an environment, and there's a representation of some location in that environment called a place cell. And when the animal's there, that cell fires, right? Well, there's other cells that fire here and here. So as the animal approaches, it's not as it approaches. This one first comes in phase with the base frequency. So at first, before you even got to this one, it's saying, oh, this one's active. And then as you actually get there, this one becomes in phase, and this one's active, meaning it's coincident with this. And then after you pass it, once you're past there, this one becomes active. So it's like it's coding a series of locations in order as you're moving, and they're going boom, 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 really fast. It would be sort of like saying if you were decoding these, and you're trying to code these, you'd say, oh, I might be here, 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 I might be here. I've always thought this is likely to be a way of counting noise in path integration. I don't know if I came up with that idea or someone else did, but the idea is like you don't always know exactly where you are. So if all of a sudden this matches your sensory input, then you say, oh, I'm actually further along than I thought I was. And I have to adjust, right? It's like if you walk in the dark in your house and I'm expecting this, and I do this all the time, and in the middle, and I go to the bathroom, make sure I don't even close my eyes, and then I walk along, I should be feeling the wall edge right here. And if it's a little further than I thought, then I re-anchor myself. You can just feel it. You can just, oh, I wasn't there, I'm here. You can just feel that happening. So that might be what's going on there. I don't know if I answered your question. The question is, are these hard-coded? I think the mechanism for doing phase progression is hard-coded.
But yeah, there's like theories about how you can convert almost like from a firing rate code to a phase code, where like you can imagine if you have like remind myself saying this. Yeah, like if this is like membrane voltage here, and this is time. And imagine you have like two neurons that get a lot of inhibition, which brings them down to like a fairly common membrane potential. Because I think the way conductance works in synapses, I'm forgetting all of this now, but generally like there's only so negative that they can go below the like kind of zero before essentially the sign of how a voltage gate is acting flips. And so they'll basically they can almost like rest at this very negative value. And then if you imagine, okay, these are two different neurons and they're receiving different amounts of firing rate input. One of them is receiving more So its membrane potential is gonna rise quickly. This one is receiving less. Its membrane potential is gonna rise less, slower.
So these are not spiking yet, these are just.
These are not spiking yet, but
Okay.
at this point, they reach the spiking threshold. Then the one that was getting the higher firing rate input is gonna spike earlier. And this will actually be proportional to
Oh, I see.
how different the
Oh,
fire
I
rates
see.
are.
Oh, that makes sense, yeah, that makes
Um,
sense,
but,
yeah.
So that relates more to rate encoding or like if you're getting.
well, it would be kind of using rate coding as a basis.
To switch to a phase encoding,
Yeah,
because once you spike,
Doesn't
there's
matter.
no more rate. The rate spikes are spikes. There's
Sorry,
no
I'll
more
be
rate.
right back.
I mean, there are some people who believe that information is encoded in the intervals between spikes. I think the evidence is really weak. They like it because you can encode a lot of information that way. But I think with, rate encoding, the problem with rate encoding is a simpler version of that. It's just saying, what's the rate at which this is happening? The problem with rate encoding, it's very slow. Like between one spike and the next takes some time, and you don't have any rate until you got the second spike. So, it's really slow. Anyway, this is interesting. This is sort of like an integration. It's an integration of input that then converts to a spike at a particular phase. in a
Yeah.
sense, or time, right? It's interesting.
and, yeah, and then for what it's worth, I guess there's, there's another one, which is like polycriny, which is,
That one, I don't know that one.
that it's this one that this guy Ezekevich in 2006 he wrote a lot about, but it's because with this phase code, this is kind of a synchronous code, like it's saying that these neurons are synchronous at this point, which is why they're or like
They're synchronous with the.
they're synchronous at the point like relative to the phase at which they fire, they work together.
Well, they're all at the same frequency, right?
Yeah, but as in like isn't it like we have a you know let's say these neurons all fire with respect to this like background oscillation or whatever. But we're gonna say these like neurons are kind of firing together. These are encoding something together because they spike at the same
Right,
time
right,
relative
right,
to that
right.
phase.
It's,
So in that sense, it's like synchronous.
okay, it's synchronous only in the phase peak,
Yeah,
okay.
but
Well, the phase peak actually relative to the base frequency, because
yeah,
all those cells in those mini columns will be firing at the same rate, and you'll have the same. They'll all be firing the same rate. It's just like relative to the base frequency, which ones are maximum.
are actually there, yeah. But then who's can I guess if I join the Zoom link, I can say.
You want to unplug
Share
my
screen.
Or is it?
Uh,
You can
there
just-
might be.
You want to just plug
Like
this
that
in?
thing.
Oh yeah, if I can just plug that in.
That's fine.
So polycrinic code is kind of where there's some offset in when the neurons are firing. So yeah, this is okay, imagine you have these three input neurons, B, C, and D, and the output neurons, A and E.
Yeah,
And so if they all fire at the same time,
what's all B, C, and D or?
B, C, and D, so,
Yeah.
oh yeah, sorry, time is along the x-axis.
Are these milliseconds, these are axonal delays?
Yeah, this is the
Oh,
axonal delay thing,
okay.
which I appreciate has issues, but yeah.
Yeah, okay.
But anyways, I just thought while we're talking about neural
Okay.
codes, I thought it'd be interesting to mention. Yeah, and then these are the different neuron IDs here. So these fire at the same time in this instance, and then these arrows are kind of indicating the conduction delays associated with their spikes, because the spikes is like an all or nothing instance, and there are, like, depending on the axons you look at, there are relatively large conduction delays associated with them.
Some,
Some.
right, some are very fast.
Yeah, and then, but anyways, if these neurons all spike at the exact same time, then, you can see that these signals arrive at different times. And
Right.
again, some neurons, not all, but some neurons are very sensitive to the exact time at which inputs arrive. But if they fire with this particular pattern, where the D neuron fires first, then the C, then the B, and with this precise delays, then they all arrive at A simultaneously. If they fire with a different pattern, they all arrive
Yeah.
at D simultaneously.
So what is the argument, though, is this?
So this is like an extremely rich code in that, like, so you can imagine It would start looking something more like this, where
But it is rich, but is there evidence of this actually happening?
I think similar strength to like phase code outside of the entorhinal, or
Okay.
sorry, the hippocampal complex.
Yeah.
So like, I think it's definitely speculative, but I mean, in theory, neurons can do it. And like there's also what's called myelin plasticity, where myelin is like the fatty sheath around neuron axons, and that can change over time, which can change the conduction delay of neurons. So,
Yeah,
but
I could see that, oh, it's not worth getting into it.
but yeah, it could create some really cool kind of interesting.
This is a little bit like, people who argue for, spike timing
STDP.
delays, right? Like, oh, but.
Yeah,
And it's like very high capacity, we like it because of that, mathematically it's nice, but I don't know if there's anything to do with brains,
yeah,
you know?
yeah. I think it's potentially not very robust to noise, depending on.
And then how would you encode it? I guess you're saying maybe the myelination would change and you form memories by changing the myelination.
Yeah, so
It's possible. I wouldn't say it's not impossible, but that's a pretty slow learning
Yeah.
process.
yeah, so it was actually something I kind of explored in my thesis, because this was like a way of encoding that my professor was a big fan of, and
Oh,
like the
sorry.
two main. No, that's all right. I mean,
Sorry.
I am equally skeptical. Like a part of like one of my chapters was literally like looking at, okay, could you realistically encode something, and I was actually trying to avoid myelin plasticity, and so just say, okay, can STDP, given input synapses, select the correct conduction delays, given like, so we've known, or like we know from certain experimental studies, like what the distribution of conduction delays looks like,
Yeah.
and it's this kind of like log normal distribution,
Mm.
where it's like kind of tailed, so it's like most, as you say, most connections are quite short and fast, but there are some, even in like nearby areas of cortex, that can be up to like, I think it's like 20 milliseconds,
Yeah,
like if they're like very fine.
yeah,
But anyways, and then, yeah, you get a neuron sees a bunch of input synapses, and then STDP, which is very time sensitive, this is like the learning rule, might select the right ones. But yeah, what I found was like there was this narrow space where like there's all these different kind of parameters that affect, like there's the time constant of STDP, there's the distribution of axonal conduction delays,
yeah.
and all this stuff, and there's this narrow region where it might just work. Oh, and there's the sparsity of
What am I
connections in the
gaining
brain.
from this? Because if I'm not learning,
So you are learning.
but what am I
So
learning?
you're selecting some patterns that you become
Right,
highly
but,
specific to.
fine, but the actual delays don't really matter, right? I learned a different set of patterns, but I mean,
Well, it matters in the sense, it's kind of learning in the same way that like an SDR is like,
right, but I don't,
but the difference is that like, you can basically reuse, like the same neurons it's primarily a capacity thing. So like the exact same neurons could be active, but if they're active in different points in time, they will encode a different pattern. So it's like,
right.
it's an SDR where you kind of add an additional dimension of like,
Right, on the receiving
capacity.
end, I don't know any of this stuff. On the receiving, I don't know what your actual delays. I just say if it matches, it matches.
well,
Great.
but you know which one in the same way that like a neuron that's sensitive to a particular SDR doesn't like care what neurons, it just knows, okay, I've received, like my dendritic segment is active.
I get coincident
Yeah.
inputs.
Yeah, so
if
this
there
is
was-
coincident inputs.
But if there were delays in those coincident inputs, I don't know that. I just say it's still coincident. So that's the pattern I'm gonna learn.
But so you only respond when they arrive in that narrow window.
Right.
So
right.
you're sensitive to just
Right,
more
but
specific
I'm always sensitive to that. The SDR, the learning rule in the dendrites that we know
Yeah,
is it's very a small window. Things have to arrive.
yeah,
And so imagine I'm going between layer 4 and layer 6A, and there's millions of fibers going back and forth. I'm trying to learn these associations between them. I guess no, that's not a good example. I'm just saying I can't take advantage. I'm not really. Yeah, the same neurons could represent. It would be a different time, but I can't take advantage of it. It feels like it is what it is. It's like I'm trying to recognize.
I mean, you can, I guess, just in the sense that like any neuron that's receiving, let's say an SDR, like we no longer, like if we're concerned about the capacity of the SDRs, we don't have as much of an issue in that like, okay, this this set of neurons, they encode this SDR, they've already been used. And it's like, yeah, but with what temporal delay relative to one another,
Right,
because
right.
that's like a totally
Well,
different
but-
code
but-
if they have a different temporal
But if an
delay.
SDR, I have no capacity problems. I guess I'm just.
And then, okay, yeah, and then I guess the second potential benefit is like,
If I'm the sending act neuron,
yeah,
and I can encode different delays,
Yeah.
then I can encode different things. But if I'm just relying on the delays that exist in the neural pill, and they're just what they are, I can't control it, and the receiver can't control it, so they're kind of invisible, like I can't really use them. It's just, it feels like a little weird, unless I have some control over it.
and then I guess the other potential advantage is like, if your input signal is somehow temporal, then that can influence
Maybe.
how you were receiving that information. And so again, that was what I was trying to explore, was like
Yeah.
whether you could do something kind of like capsule networks, where like as you move a feature in a receptive field, that changes like a spike
Yeah.
time,
Yeah.
and then whether you get some coincidence. Anyways, it's
Let's bump this out again. It's interesting. Vivian's back, so she suggested maybe some future time we talk about this, because encoding is sort of like key to everything. By the way, for the newcomers here, we don't do any of this spiking stuff right now. We're just using real numbers. And there's been a debate whether we need to, right? So maybe it's all useless. But it's really nice to know what's going on in the brain, because I find it's generally not useless. Even if you don't do it this way, to know what are the things it is representing, how does it do, the different types of information you can represent, is very, very helpful. So there's sort of a spectrum here, right? You have a spectrum like one cell firing rate, very limited. You might have the mini column hypothesis, which is essentially a limited number of things, or mini columns, but you could, it's only a few of them, and and so it's not a rate encoding as much as it's it's like a place code, if you will, like which three of these is active at one point in time. That's more capacity than this, but not a lot. But it's you're not relying on one cell, which nothing really works with one cell. And then we talked about really sparse stuff, but maybe there's some things in between. Maybe there's some things in between that are between non-sparse and super sparse. And the idea that maybe they change over time, I don't know. I mean, there is a question about that. And then the whole phase thing is another play on top of that. So maybe there are in between representations. We do see a lot of variety of density in the cortex, right? You can see very, very sparse representations where they see a cell fire 200 times in 30 minutes. I mean, that's not much. That's like, you better keep your eyes open, you'll miss it. It's super sparse to things that are much less sparse than that. So there could be, and we've talked about ways you could have multiple ones, less sparse and a variety of sparsity, and this idea you just brought up could be another one along those lines. Anyway, that is an interesting question to collect data about this specifically, and maybe come up with some thoughts about it, further thoughts about it. I don't know. One thing about the nice thing about our neuron paper is we introduce a mechanism where you go between two of these modes. You go between those not so sparse mini column representation and a super sparse SDR. So the SDR is always, the same. This representation, which is not so sparse, has many specific sparse ones that are all equivalent to one of these. So in some sense, you are going between. You can go between these. It's like you have two representations simultaneous. You have the mini column representation and the SDR version of that. They're really in the same cells in some sense. So in that sense, it is like a sparse and less sparse version.
When we say it's 2% sparse, is that in the mini columns or in the SDR?
That would be the SDR version. This is like 3 out of 10. That's not very
That's even
sparse.
sparser.
No, this is not sparse at all. This would be like 30%, right? 2% is, I have 1 million cells and 10,000 are active. So,
But the whole population is going to be sparser. Like all of the mini columns, if you count all the cells that are active.
well, if you count the cells, then they're sparse.
Right.
That was the whole
But
point.
the mini column itself is not sparse.
Right. So in the neuron paper, you might say you might have 20 cells in a mini column in a particular layer, right?
Mm.
That's not unreasonable, and good number. So you got 20. So if I'm essentially I'm ready, if I have, I might only have like, 10 different representations here. But for each of those 10, I can pick, 20 to the third, is that right? It's 8,000 versions, 8,000. There's 8,000 ways of representing each of these, orientations. All right, all right, yeah, about right. So, so you can get a much higher representation space by sparse bindings, right? In this case, you'd have, since if there's 10 columns, you have 200 cells here. These are pretty low numbers. It would be bigger than this. 200 neurons, of which only 10 neurons are active. So it's 200 choose, it's like 200 choose 10, but it's not quite. So anyway, that was, I think, one of the greatest things we came up with here, because it really branched between these two modes of like an input which is not unique to an input which is the same input, but it is unique. And an evidence for it is very high. So that's almost certainly correct in some form. Well, today was a day of just banging our heads against walls and speculation and going over problems we don't know.
Yeah, did we have any new ideas today?
Yeah, at least from the thalamus thing, I think we talked about like, yeah, one takeaway was maybe the competition thing.
Yeah.
Like, yeah, I think we definitely had lots of like interesting biological ideas, but in
Yeah.
terms
I put these in a file in your head of things you can come back to later when a problem comes up and you say, well, how am I going to solve that problem? And, oh yeah, that could do it,
Yeah.
It was opposed to like, oh, we solved the problem. I don't think that was the case. And we didn't do that today. Some days we do that, but not today.
So good to go over all of this again and kind of clarify the problems and questions.
Yeah, it's yeah. It's it's just like a settling process. You throw all these crazy ideas around and eventually it sorts out. Makes sense.